All right, let's get right into it. Inside every single one of us, right now, there's this constant silent war being fought against cancer. And the front line of that defense, it's not some fancy drug. It's actually two microscopic proteins, these two tiny guardians that keep our cells from going rogue. So today, we're going to break down exactly how they work. I mean, just think about that number for a second. Trillions of cell divisions. That's trillions of opportunities for something to go haywire. So, what's the secret? What's the machinery inside our cells that keeps this whole process from just spiraling into chaos? Well, it turns out the answer lies in a really brilliant security system. And that's really the key thing to understand. Cancer isn't just random, messy growth. For a rogue cell to actually become successful, it has to be methodical. It has to break a very specific set of rules, essentially dismantling the very checkpoints that were designed to stop it. And at the absolute heart of those checkpoints are what we call tumor suppressor genes. Yeah. Think of these genes as the cell's internal security guards. Their entire job is to pump the brakes on cell division, to check for any damage, and to make these life-or-death decisions that protect the whole body. Today, we're going to focus on the two most famous, most important members of this security team, RB and p53. Okay, first up is the retinoblastoma protein, or just RB for short. Honestly, the best way to think about RB is as a proactive gatekeeper. It literally stands guard at this critical checkpoint in every single cell's life, and it decides who gets to go forward and who has to wait. So here's how that works. Before a cell can hit copy on its DNA, which is the absolute point of no return for division, it has to get permission from this RB gatekeeper. And RB's default setting is always stop. It's always on. It actively holds the cell in this waiting period, which scientists call the G1 phase. So how does RB actually do this? Well, it's not just a wall. It's smarter than that. It works by controlling another set of proteins called E2Fs. Just think of the E2Fs as the keys to the engine of cell division. They're the things that can turn on all the genes needed for DNA replication. And RB's job? It's simple. Just hold on to those keys and do not let them go until the time is right.
So, what does the right time look like? Well, it's a beautifully orderly process. It all starts when the cell gets signals from the outside telling it, hey, it's time to grow. These signals activate these little enzymes that act like a key in a lock. They unlock RB by sticking a little chemical tag on it called a phosphate group. That one simple change alters RB's shape, and it forces it to let go of the E2F keys. And boom, just like that, E2F is free, it starts the engine, and the cell moves forward to copy its DNA.
Now, I know this diagram looks a little complicated, but it's really just a map of what we just talked about. See those signals coming in on the left? They start this whole cascade that's going to unlock RB. And right there in the middle, at step C, you can see that machinery adding the phosphate tag to RB. That is the literal unlocking step. And what happens right after? You see RB letting go of E2F. Now, E2F is free to launch the whole program for cell division. It's a perfect controlled chain reaction.
Okay, let's move on to our second guardian, p53. This one is so important, it actually has a famous nickname, the guardian of the genome. And if RB is the proactive gatekeeper checking your ID before you get in the door, then p53 is the reactive, screaming red alert emergency system. It's like the cell's smoke detector, radiation alarm, and intruder alert all rolled into one.
And this is a really important difference. In a normal, healthy cell that's just doing its thing, p53 is just kind of hanging out in the background, totally quiet. It doesn't get involved in the day-to-day decisions that RB makes. Nope, it's a specialist, and it's just waiting for signs of really serious trouble. And what kind of trouble are we talking about here? The big stuff. Things like DNA getting damaged from, say, UV radiation or chemicals, or a lack of oxygen, which is a massive stress signal. And, this part is so cool, the activation of oncogenes, which are genes that push for cancer. So if a rogue growth signal suddenly appears out of nowhere, p53 sounds the alarm.
This chart really lays out the two different security philosophies side by side, doesn't it? You've got RB, the everyday traffic cop, proactive, routine, preventing problems before they even start. And then you have p53, the emergency SWAT team, totally reactive, incredibly powerful, and only called in when there's a major crisis. They have completely different but totally complementary jobs.
So once p53 is activated, it becomes the ultimate judge, jury, and executioner. It can hand down two main sentences. First, it can order a cell cycle arrest.
a total lockdown, giving the cell's repair crews time to fix whatever is broken. But if the damage is just too much, if it's beyond repair, p53 makes the ultimate call. It triggers apoptosis, or programmed cell death. It basically forces the cell to commit a clean, orderly suicide to protect the greater good.
So we've got our gatekeeper, RB, and we've got our emergency alarm, p53. You'd think that'd be enough security, right? But cancer is clever, and to succeed, it often has to pull off a conspiracy against both of these guardians.
Let's just think this through for a second. Imagine a potential cancer cell finds a way to break the RB brake. Now it can divide whenever it wants, ignoring all the normal stop signs. You'd think it's won, right? Game over. Not so fast. In this is the most elegant part of the entire system. When RB is broken, those E2F keys are just floating around all the time. And that constant, unregulated E2F activity is interpreted by the cell as a rogue growth signal. Exactly the kind of oncogene activation that p53 is designed to detect. So breaking the first security system automatically triggers the second. It is an absolutely brilliant failsafe.
And this isn't just my interpretation. This quote from the source material says it perfectly. As soon as you disrupt the RB pathway, this p53-dependent program kicks in as a backup, either slamming the brakes on again or, more dramatically, just killing the cell outright. The system is designed with a backup plan.
So the big takeaway here is this. It's not enough for a cancer cell to just hotwire the accelerator by breaking RB. It also has to cut the wires to the emergency alarm by getting rid of p53. It needs two separate hits, a true conspiracy, to finally break free from the cell's control. And you know what? This isn't just a theory in a textbook. When we look at actual human cancers, this story plays out again and again. Just look at these numbers. In some of the toughest cancers, like pancreatic cancer and small cell lung cancer, the RB pathway is broken in a staggering 80% of tumors. And you can bet that in the vast majority of those cases, the p53 pathway is broken right alongside it. Now, I know this all might sound a little grim, but understanding this two-hit conspiracy is actually our greatest weapon. Because by knowing exactly how the cancer cell cheated, we can design much smarter, more targeted ways to fight back. So instead of just poisoning all fast-growing cells with old-school chemotherapy, we can now design drugs that specifically go after the vulnerability created by a broken RB or a broken p53. It's like we found the enemy's secret playbook, and now we can use it against them. This map here shows us the new battleground. It's the same pathway from earlier, but look, it's now covered with all these therapeutic strategies circled in blue. Each one represents a potential point of attack for a new drug, a specific weakness we can target. We are no longer fighting blind. We are launching precision strikes. And these aren't just science fiction. Some of these are in clinics right now. We have drugs called CDK inhibitors that act like a new set of brake pads for that RB system. There are new drugs trying to fix broken p53 proteins. And get this, there are even engineered viruses that are designed to only hunt down and kill cells that have a broken p53 system, leaving all the normal cells totally unharmed. It's incredible stuff. And this all leads to a final, really powerful thought. We're moving away from a one-size-fits-all approach to cancer. By reading a tumor's DNA, we can figure out its specific conspiracy. Which guards did it take out and how? And that lets us choose the exact weapon to exploit that weakness. So the question is no longer just, what cancer do you have? But how exactly did your cancer break the rules? And that, right there, changes everything.